Science is meant to organize the world. It sorts, measures, compares, and names things until they sit in the right place. Most results, even difficult ones, eventually do. And then there are the rare cases that do not. Not because the evidence is false, and not because the past contains something supernatural, but because the categories we rely on are sometimes smaller than reality. The first sign of that is usually not dramatic. It is a quiet line in a report, a graph that looks wrong, a sample that refuses to settle. In one genetics laboratory, the work began the way it usually does. A labeled bag, a fragment of bone, a set of numbers on a form, a burial recovered from central Italy, dated to the last centuries before the empire, when Rome was already expanding, but still officially a republic. The remains were weathered, incomplete, and ordinary in the way ancient remains often are. No gold, no inscription, no official name, just a person preserved by chance. Handed over to modern methods. The expectation was simple: Central Italy, late Republic. The DNA would be familiar, not identical to modern Italians perhaps, but still recognizably part of the same broad story. Close to other Iron Age individuals from the peninsula, close to what historians imagine when they picture early Romans, early Italians, the ancestors of a place. The first sequencing run produced enough data to begin, and then the comparison began. At this stage, the analysis is often mundane. The sample is placed into a larger reference set. It should drift toward one cluster rather than another. It should sit where its context says it belongs. But this one did not. It did something that makes geneticists pause. It fell outside the main cluster of comparable ancient Italians in the dataset, not slightly, clearly, as if it belonged to a different map. For a moment, it is tempting to call that a no match. In practice, that phrase does not mean the DNA is inhuman. It means the sample does not match what the lab expected it to match. It does not resemble the local baseline they have built their models around. It does not settle into a neat category, and in a field where error is always possible, the first instinct is caution. Contamination is the usual suspect. Modern DNA can seep into ancient material through handling, through soil, through equipment, through time. Even the cleanest laboratories treat contamination like a shadow that never fully leaves. So the team did what careful teams do. They checked authenticity signals. They looked for patterns consistent with ancient DNA rather than modern DNA. They tested the proportion of modern contamination. They repeated key steps. They did not rush to interpret a single strange plot. The result held. The DNA looked ancient. The contamination, by their estimates, was negligible. And still the sample sat where it should not have sat. It was shifted toward what geneticists broadly call the Eastern Mediterranean, a direction on a genetic map that carries centuries of history beneath it. Because if that shift was real, it raised a question that feels simple but isn't. Who exactly was in Italy before the empire? And what does it mean to say someone is Italian in the first place? To understand why this is unsettling, you have to rewind. Long before modern nations existed, the Italian peninsula was already a meeting place. Its geography almost forces it. It stretches into the Mediterranean like a bridge. It faces the Balkans, the Aegean, North Africa, Iberia, Central Europe. Trade routes cross its coasts. Sea routes brush its islands. Even its mountains do not seal it off completely. For centuries, the popular picture of Italy's origins has been uncomplicated. The Romans came from Italy, expanded outward, and then became a symbol of Italy itself. The peninsula becomes the center, and everything else becomes the periphery. When people imagine origins, they often imagine roots, something that starts in one place and stays there. But genetic history does not work like that. It works in layers. Before Rome was a city of marble, Central Italy was a patchwork of communities. Etruscan-speaking groups, Latin-speaking groups, Greek colonies in the south, Phoenician networks along the sea, local societies with older histories beneath them. The Iron Age Mediterranean was not isolated. It was connected in ways that can be easy to underestimate, because the modern mind expects borders where there were none. This matters, because it changes what unexpected DNA truly means. An Eastern Mediterranean signal in Central Italy does not require a single dramatic event. It can arrive through trade, through mobility, through war, through resettlement, through intermarriage. It can arrive slowly, in increments, until it is visible in the dead. And most importantly, it can arrive before the moment we instinctively point to, before emperors, before the empire. The lab result hinted at that. It suggested that the genetic shift so often associated with Imperial Rome may have begun earlier, not fully formed, not uniform across the peninsula, but present, already moving, already there. This is where the story becomes delicate, because there are two ways to tell it: a reckless way and a careful one. The reckless version says the origin story is false, that the people were never local, that everything collapses at once. It is clean, it is simple, and it is often misleading. The careful version says something quieter. It says the origin story is misleading because it pretends the past was tidy. It pretends lines were straight. It pretends people stayed in place. But the evidence suggests they did not. Even within the same region, within the same century, individuals can look different genetically. Some sit inside the local cluster, some sit outside it. Some appear to be outliers, people whose ancestry does not match the majority around them. And the moment you admit that, another uncomfortable idea follows. If one person can sit outside the expected cluster, how many more were there? How many have not yet been sampled? How many will change the map again? The deeper you go into Italy's past, the more the idea of a single origin begins to dissolve. In the very long view, Europe itself has seen major population turnovers. Genomic research suggests that hunter-gatherer groups were replaced or absorbed in southern Europe around the last Ice Age peaks, followed by the spread of new ancestries after around 14,000 years ago, pushing northward and reshaping much of the continent. That is not a footnote, that is the foundation. Then, later, the arrival of farming brought another shift. New lifeways, new animals, new crops, and people moving with them. And then, in later prehistory, the arrival of steppe-related ancestry appears, again, reaching into the Italian peninsula by the Bronze Age. At each step, the illusion of a single unbroken line becomes harder to maintain. By the time you reach the Iron Age, the Mediterranean is increasingly connected. Seafaring improves, settlements expand, colonies are founded, networks form, and genetic heterogeneity becomes not an exception, but a likely condition of life in many coastal regions. This matters because it reframes the Roman story. Rome did not expand into a static world. It expanded into a world already moving. And then Rome in turn intensified that movement. The empire is often described as a system of mobility, not because everyone traveled freely, but because soldiers, merchants, administrators, enslaved people, and displaced communities moved across enormous distances under one political framework. In genetic terms, that kind of system tends to leave traces, and it does. Studies of genomes in and around Rome describe a shift toward Near Eastern ancestries during the imperial period, accompanied by striking diversity between individuals. Then later, as political structures change and new migrations occur, the ancestry profile shifts again. What the lab results suggested, quietly, without drama, was that parts of this pattern may have begun earlier than we instinctively expect, that the genetic story did not wait for an emperor to start, that it was already underway in the late Republic, and yet, even now, that does not fully explain what the lab saw. Because Eastern Mediterranean is not a single place. It is not one people. It is a broad label, used partly because the data cannot always distinguish finer origins. Differences between regions can be subtle in the genome. Reference samples for certain areas and periods are still limited. Southern Italy, in particular, may be underrepresented compared to central sites, meaning a signal that looks unfamiliar might actually reflect internal movements along the peninsula from places we have not yet fully characterized. In other words, the no-match feeling can be created not by the DNA itself, but by the gaps in our comparison sets. This is one reason why scientists tend to hesitate, because a surprising result can mean several things at once. It can mean the person was a migrant from elsewhere. It can mean the person's parents were migrants. It can mean there was already gene flow into the region before the period we associate with it. It can mean the reference panel is incomplete. It can mean the statistical model is sensitive to the available data. It can mean occasionally that technical artifacts have nudged the result. And those technical artifacts are not imaginary. Ancient DNA is often fragmented and low coverage. Short fragments can be more difficult to map accurately. Analyses can be affected by reference bias, where reads matching the reference genome align more easily than reads carrying alternative variants. Even small biases can slightly influence estimates of affinities and ancestry proportions if they are not assessed carefully. Good laboratories know this. They check, they filter, they test. But uncertainty does not vanish simply because the process is careful, it changes shape. Instead of asking whether the data is real, the question becomes what the data actually means. This is where interpretation begins to diverge. Some researchers emphasize the breadth of mobility in Roman and pre-Roman systems. They see outliers as reflections of real human movement in a connected world. Others focus on sampling gaps. They argue that what appears unusual now may appear ordinary once more regions and periods are fully represented. And some insist on a further point, which is perhaps the most difficult to hold in mind. DNA can describe ancestry. It does not define identity. It does not tell you what language a person spoke. It does not tell you what they called themselves. It does not tell you how they were perceived by those around them. A person with ancestry shifted toward the Eastern Mediterranean could have lived their entire life in Italy. They could have been socially Roman, legally Roman, culturally Roman. They could have been buried according to local customs. They could have belonged completely to the place they died. The genome does not confirm belonging. It traces biological history and nothing more. And yet, the result still unsettles, because it disrupts a habit of thinking. People prefer a clean origin, a single starting point, a story that runs in a straight line. Italy, perhaps more than many places, encourages that instinct. A peninsula with deep history, a civilization often treated as foundational, a modern identity that draws from ancient symbols. To discover that the genetic reality is layered can feel like contradiction, as if the origin story was constructed rather than discovered. But the truth is quieter than that. The story is not false because it was invented. It is incomplete because it was simplified. The peninsula was never a sealed space. It was a corridor, a shoreline, a network of routes and crossings. It gathered people, lost people, absorbed people, and changed over time. Even when political structures appeared stable, the demographic reality could shift beneath them. And that is the quiet implication of that first unexpected result. Not that the sample was impossible, not that continuity does not exist, but that the frameworks used to describe the past are often narrower than the past itself. The most unsettling part comes after that, because once you accept that one individual can sit outside the expected pattern, you begin to consider how many others remain unexamined, unsequenced, uncompared, still part of the ground. Each one capable of adjusting the story, not through speculation, but through evidence. And as more data is collected, the pattern does not necessarily become simpler. It often becomes more complex, more regional, more conditional, more precise, but less tidy. Which means the feeling of no match may never fully disappear. Not because the past is unknowable, but because it is unevenly preserved and unevenly studied. Some populations are well represented, others remain largely absent. Some regions are mapped in detail, others are still largely blank. So a result that does not fit can reflect both presence and absence at the same time. Presence in the data, absence in the comparison, absence created by time itself. In the end, Italy's genetic history does not point to a single alternative origin. It does not replace one simple story with another. It reveals a long sequence of movements, interactions, and changes, some gradual, some abrupt, many still unresolved. The only thing that truly disappears is the idea of a single origin. And perhaps that is what makes the story linger. A person buried in central Italy nearly 2,000 years ago produces a result that appears out of place. The laboratory checks again. The data remains consistent. The map shifts slightly. And somewhere within that shift, a quieter thought emerges. If even a place so often imagined as stable contains this degree of movement in its past, then perhaps the origin stories attached to any place are not fixed histories. They are simplified versions of something more complex. And when tested carefully, the past does not always conform to those simplifications.